Good morning or good afternoon, depending on where you are in the world. My name is Dr. Borja Alvaro Barcenilla, and today I will be talking alongside Harrison Cocker and Rachel Rivero about growing beyond lessons from the moon for Earth's future farming. This is a webinar hosted by the Moon Village Association Lunar Agriculture and Sustainability Working Group. Part of this work that is presented today has been funded by NASA and the NIH. First of all, why do we need plants in space? We're going to need plants as a food source. Within the next decade, humanity is going to attempt the colonization of the moon, and for that, astronauts will have to grow their own food. As well, we need plants for oxygen generation, carbon dioxide removal, and mental health. When you talk or hear the astronauts regarding the happiness and the joy that they get from working on plant experiments on the International Space Station, it really explains what plants are going to be very beneficial for mental health purposes on future colonization attempts on the moon.
Favorite experiment on the International Space Station?
Toby, great question. My favorite experiment was the one that I got to eat. It was us growing hatch chili peppers in our plant habitat, and some of them got frozen to go back to the ground, but many got to stay with us and we ate them.
Station Huntsville on 2, we have video in Node 1. Look at those chili peppers.
What we're doing
witnessed or been a part of.
You're going to do fluid physics one day, you're going to do medicine or biology the next day, so that's pretty special for us every day. I think one that we actually really like that's ongoing right now in that very module, it's called Plant Habitat 4, and we're growing some plants on board the space station because when we venture further and deeper into space, we'll need to be able to grow our own food. So we have to study this on board the space station, and by doing so, we also identify how to make plants maybe more resistant to difficult conditions where water is scarce, when they're liking what they usually get from the natural environment. Because as you can imagine up here, it's actually difficult. There's no soil, you have to water the plant, everything becomes much more complicated. But nevertheless, we were able to, with the help from the scientists.
So on today's talk, first of all, we're going to talk a tiny bit about the history of most of the plant experiments that have taken place in low Earth orbit as well as on the moon. It's actually very interesting that the first organisms in space were especially developed strains of seeds, as they were termed, that they were launched on July 9th of 1946 on White Sands in New Mexico on top of a B-2 rocket that you see over here on the right. The first seeds that actually launched into space and they were successfully recovered were maize seeds that launched on July 30th, 1946, which were soon followed by rye and cotton. Later on, in 1971, 500 tree seeds that included pine, sycamore, sweet gum, redwood, and Douglas fir were flown around the moon on Apollo 14. Some of those seeds, they came back to Earth and are currently grown in different parts of the world. Here is a picture of one of those descendants of the moon tree seeds that are being currently taken care of at Texas A&M University Gardens. We go on. In 1982, the crew of the Soviet Salyut 7, which is depicted here on the right, space station, conducted an experiment prepared by Lithuanian scientist Alfons Merkis that grew some Arabidopsis using Phyton 3 experimental micro greenhouse apparatus, and they became the first set of plants to flower and produce seeds in space. Later on, there was a Skylab Rise study, in which you can see a picture over here on the right. And then the SVET2 space greenhouse successfully achieved the first seed-to-seed plant growth in 1997 aboard the space station Mir. You can see over here the SVET2 greenhouse on the left and the Mir space station here on the right. As we move on through the years, the BPS, or the Biomass Production System, that is featured over here on the right, was initially designed for shuttle and space hub, but was later modified to fit in the International Space Station, and it contained 2 experiments on the ISS in 2002 during Expedition 4. Since 2003, Russian cosmonauts that are on the station have been eating half of their crop, while the other half goes towards research. And then Expedition 44 members became the first American astronauts to eat plants grown in space on 10th of August of 2015. In this case, they ate lettuce that they grew during their expedition. If you are further interested in some of this space plant history, you can further get more information from astrobotany.com, from the great podcast Gardeners of the Galaxy, as well as from the website Salad. So far, what have we learned from plants grown on space? We have seen that plants do display minor physiological changes. Flowering is achieved in many species, even though in some cases it has been delayed. In some cases, there has been denser root hairs observed, as well as sometimes asymmetric shoot architecture. One of the major common molecular themes that we observe across all plant species in space is signs of molecular stress, and there has been experiments that describe increased anthocyanins, increased reactive oxygen species and oxidative stress, increased genome oxidation, as well as cell wall increase. So, even though we have seen minor physiological changes in plants, there is a very important question that we need to tackle, and it is that we still do not really know if plants are going to be long-lasting in space. There are some key indicators of organism survivability and prosperity, such as DNA damage markers, epigenetics, mitochondrial dysfunction, but the most common one is telomere homeostasis. But first of all, what are telomeres? Telomeres are DNA repeats, TTT-A-GGG, at the end of the chromosomes that they do not code for protein, and their function is the protection of the whole genetic material. Every organism has a different telomere length set point, which maintains the rest of the genome protected. In Arabidopsis, that range is between 2 to 5 kb, in mice between 20 to 100 kb, and in astronauts between 5 to 15 kb. Mutations in telomere components and telomeres can lead to either telomere shortening or telomere lengthening. Telomere shortening is one of the hallmarks of senescence, and telomere lengthening has been associated with many different cancers. The selection hypothesis dictates that organisms with short telomeres have lower chances of species prosperity. It's important to say that environmental as well as oxidative stress, meaning increased reactive oxygen species, can have an impact on telomere length as well as telomerase activity. Telomere lengthening has been observed during human spaceflight, and it has been tested in the famous twin study experiment, as well as a 6-month astronaut missions, and more recently on Inspiration 4. Experiments revealed that telomere length increased during spaceflight, and this telomere lengthening was independent of the time on the station, as it was very rapidly observed in astronauts that were very little time in space. One of the major hypotheses of why this telomere lengthening could be happening is due to increasing telomerase activity. However, no telomerase activity were measured for in-flight organisms, and the human telomere length directly correlates with telomerase activity. Astronauts as well experience increase in ROS and genome instability. So the first question that we have is, whether plant telomeres will suffer the same fate, meaning plant telomeres will as well get elongated in space. To test for that, we measure telomere length of both shoots and roots of the plants grown on the International Space Station compared with plants grown on the ground at Kennedy Space Center. We saw that compared with the control samples, flight shoots had the same telomere length compared with the ground, leading to no statistical change in telomere length between space flight and ground control, a similar response for roots flown aboard the International Space Station. On the other side, we observed that oxidation of the plant genome increased during spaceflight in both shoots tissues as well as in roots, compared with the ground controls. We recently launched another experiment to the International Space Station aboard SpaceX 32, named Apex 12, in which we're going to see whether different genotypes and plants enhanced to reduce the levels of ROS are able to display less genome oxidation compared with the ground control samples.
So, we have seen that plants, despite their increased genome oxidation, they should be able to be long-lived on low Earth orbit, as plant telomeres remain stable aboard the International Space Station. So now the big question is, can we use those plants as a source of food for astronauts?
The work I'm going to present here was recently submitted and is under review. As you will see, this strategy is in line with the work being done by the last working group. The primary question we were trying to answer is how can personalized dietary strategies counteract space-induced nutrient deficiencies and health risks? This is particularly of importance for extended missions beyond LEO, where resupply is not an option. To explore this, we combined two different studies of lettuce grown in LEO, where the nutritional content was analyzed, and looked at that potential effects on astronauts related to nutrient deficiencies, and then 3 different astronaut gene expression data sets, and looked at how modulation in mineral and nutrient pathways can also have negative impacts. This allows us to consider potential countermeasures and considerations for these long-duration missions where growing food may be necessary. These 2 studies for plants are lettuce grown on the Tiangong-2 Space Lab and combined 3 lettuce experiments grown in veggie on the International Space Station. When we combine all of this data, there is not a reduction or increase in the mineral content. However, when we look at each component individually, we see some changes. Particularly, there is a reduction in calcium and even an increase in minerals like sodium here. To get a closer look at this data, we pulled out some key minerals associated with diseases. I want to especially point out calcium, where there is quite a decrease on the Tiangong grown lettuce. We all know that bone density loss is a serious concern in microgravity, but we also see this similar but not as drastic trend in magnesium and iron, highlighting other risks that may be associated with spaceflight. Compared to the Tiangong, the ISS calcium is reduced but not as drastic. And while astronauts may not eat a kilogram of lettuce a day to get that daily value, these reductions are important to consider as adding in certain calcium-rich foods may be required. Next, we used 3 astronaut RNA-seq studies with the goal of understanding how nutrient modulations may correlate to what happens to the body in spaceflight. First is the famous NASA twin study. This looked at one twin brother on the ground and the other brother in space for 30 days. We also have some single-cell resolution data for this study. Next, we have the SpaceX Inspiration 4 study, which looked at 4 civilian astronauts that were in space for 3 days, comparing pre-flight and post-flight, also with single-cell resolution. And third is the JAXA's cell-free epigenome study, which looked at 6 astronauts in space for 120 days and covered all 3 collection time points. This study is bulk-seq using plasma cell-free RNAs. We then developed a pipeline for analyzing nutrition-relevant changes in response to spaceflight. We first identified nutrients and minerals of interest. We then determined associated genes using the IEM-base and KEGG database. We then mined the data using the SOMA browser, which contains all of the studies I discussed on the previous slide. Then the idea is that we can use this information to discuss potential associated diseases as well as potential countermeasures, as we want to see if gene expression changes in response to spaceflight may further contribute to nutrition-related health risks. The individual biochemistry of astronauts adds another layer of complexity to addressing nutritional challenges. We have seen decreased calcium content in crops grown in microgravity, which could exacerbate calcium-related health complications experienced by astronauts. Given this issue, we analyzed the biochemistry related to calcium provided in the twin study. We see very clearly in the teal color that urinary CTX increases in flight and returns to normal post-flight. We also see similar patterns in urinary calcium and osteocalcin, as well as a modest increase in bone-specific alkaline phosphatase with a slight decrease again to post-flight. These changes suggest that there may be increased bone turnover during flight with a post-flight return towards baseline levels. Then when we look at the KEGG pathways associated with combined DEGs across all studies, we see a clear relationship to calcium signaling, including involvement of the important parathyroid hormone synthesis. Another key point that we will return to later is changes in mineral absorption, which we may see correlated to leaky gut. In summary, combining what we know about calcium deficiency in plants with what we see in spaceflight, if long-duration missions of astronauts were to eat space-grown plants, these deficiencies may exacerbate dysregulation of calcium-related processes, leading to risk of health problems. Next, we looked at calcium-related differential expression using the list of relevant genes I mentioned in the pipeline. We see the twin study having the most unique, perhaps not surprising given that it was the longest study. There was also a shared group of 8 genes among all studies. These include aspartate beta-hydroxylase, which misregulation could contribute to hypocalcemia and deficiency, as well as phospholipase C gamma 2 involved in osteoclast development and therefore bone density. We can then take advantage of the single-cell data to look at some specific cell types. We clustered these into broader categories, but you can see we still have more cell types available in I4. Nonetheless, it still provides the opportunity for comparison. Based on a specific look at the individual cell types, we see overall that some monocytes suggest altered osteoclast activity, while T cells may influence bone remodeling, along with general B cell dysregulation. Overall, this represents the potential for weakened immune function and bone loss. Along with the changes we have discussed, we also see alterations of mitochondrial and calcium-related genes in response to spaceflight when looking specifically at the I4 and twin studies. This, of course, is not surprising given the well-characterized oxidative stress known to be exhibited in response to spaceflight. It is also well linked to calcium signaling. Calcium is crucial for mitochondrial function, ATP synthesis, and ROS regulation, with some genes listed here with known involvement. Therefore, we wonder if changes in calcium levels in the body may contribute to this known mitochondrial dysregulation and play a role in spaceflight-induced oxidative stress. We hope that these types of analyses can provide insights into potential countermeasures that may be established to address the risk of nutritional deficiencies and their consequences in space. One is to simply supplement the diet with foods known to be higher in certain minerals, such as calcium. All of these crops here have been grown in space, with soybeans and dill having fairly high levels of calcium. Next is biofortification, which has been studied particularly in lettuce to increase nutrient content. There is also bioproduction, such as the work using lettuce to produce parathyroid hormone, which you may remember was shown as modulated in the KEGG pathways previously shown. And lastly is just supplementation, perhaps the most straightforward but not necessarily viable for extended missions where we are looking towards Earth independence. But another concern is, are these nutrients actually going to be properly absorbed even if we can get enough? It is established that spaceflight may induce leaky gut syndrome. This is perhaps connected with the mineral absorption that we saw in the modulated KEGG pathways. In simulated microgravity, there is a loss of immune cells in the bowel wall compared to the control. Furthermore, the published analysis of the JAXA study has already identified differentially expressed genes that are crucial for maintaining gut barrier integrity. A reanalysis was done for our work, showing an association with changes in both mineral and vitamin absorption function. Therefore, it is possible that spaceflight could be causing altered intestinal permeability that may further nutritional deficiencies. Proposed countermeasures to this are restoration of the intestinal barrier by protecting epithelial cell-to-cell junctions and regulation of immune cell function to prevent abnormal response to normal gut flora.
None of this work would have been possible without the extreme hard work of all of these researchers that you see in this picture over here. There has been as well great collaboration between NASA Gene Lab, Analysis Working Group, and BioAstra. So now that we have seen what plants have to offer on low Earth orbit, we're going to go towards the moon and see if plants are going to be able to grow and if we're going to be able to eat those plants on moon. So in 2022, Annalisa Paul and her colleagues were able to grow plants in genuine lunar regolith brought back from Apollo 11 mission, Apollo 12, and Apollo 17 using this system that you see right here. The plants were able to grow, they were able to germinate. However, looking at the transcriptome analysis of the different Arabidopsis plants grown on the different Apollo sites, they displayed increased reactive oxygen species response, salt, metal, as well as heat. So plants are indeed stressed. So plants have been able to grow on lunar soil. They displayed transcriptome analysis of higher res, but they were able to grow and germinate. This speaks to the resilience of plants to stress. However, the other question that we have if we are to colonize Moon is whether we can have different plant generations live on the Moon. So for that, we need to check physiological and molecular markers of fitness. And what better way to do it than to check a plant telomeres on plants grown on lunar regolith simulant. So looking at different Arabidopsis thaliana grown on lunar regolith simulant across different generations, we can see that visually the plants are smaller as the generation progresses, such as the G2 lunar regolith simulant grown plants are smaller compared with the G1 and way smaller compared to Earth. You can see this by decreased rosette area as the generation progresses, as well as decreased biomass as the generation progresses, as well as the stalk height of all of those plants. So indeed, G2 lunar plants display worsening physiological defects compared with G1 and way worse compared with Earth-grown plants. So we question if there was an increased oxidative stress that could account for the progressive shortening in size in lunar regolith. And indeed, we observe an increase in ROS in lunar growth plants. So I just explained you in the introduction that oxidative stress can lead to telomere length changes. However, as I previously shown you, Arabidopsis telomeres are actually remarkably stable under different stressors, and Arabidopsis telomeres did not change when they were flown to the International Space Station. However, and in this case, what we are observing is a decrease in telomere length across the generations of around 500 base pairs. So this is leading us to hypothesis that the excess genome telomere oxidation can lead to erosion, which will lead to decreased survivability. So obviously, the big caution that we need to see here is that genome integrity will be compromised with continuous propagation in lunar regolith, as we have observed. So we observe this telomere shortening and elevated genome oxidation not just in one regolith simulant and in just one Arabidopsis ecotype. We actually did observe this phenotype if we are able to grow HOB-10, a different Arabidopsis ecotype that comes from Sweden, we see that using different regolith simulants, we see as well a telomere shortening happening in the plants. As well, we observe the same phenotype in Arabidopsis thaliana Pro Zero ecotop that comes from my native land of Spain. We see that compared with the earth soil, telomere shortening happens with with regolith simulant LMS1 as well as LSP2. So another word of caution, genome integrity might be compromised in different ecotypes and different regolith types. We just did not observe this phenotype in Arabidopsis. We also observed it in Brassica oleracea when they were grown on lunar regolith simulant. You can see that lunar grown Brassica oleracea was smaller compared with its Earth control, as well were the telomeres of this plant that were statistically significant shorter compared with the ground control. The question that now we have is what is this cause for telomere shortening? pH, heavy metals. We know that the regolith has a different pH as well as the presence of heavy metals, that all of these could impact the overall plant growth. Indeed, we observe that plants grown on regolith simulant accumulate aluminum and copper, which is known to be toxic to plants. We observe this not just on Arabidopsis grown in different regolith simulants, but as well we observed it in Triticum as well as in Medicago. These plants are absorbing these different heavy metals. Interestingly, we observed that Arabidopsis thaliana that were grown on MS plates with aluminum or copper displayed telomere shortening, which explains that this telomere shortening could be due to accumulation of the heavy metals on the plants. Telomere shortening observed from regolith might be then due to the toxicity of absorption of these heavy metals. So then the question arises, what were to happen if you were to eat these plants, especially since the daily toxicity levels of copper and aluminum for humans are 10 milligrams and 50 milligrams respectively?
Hi everyone, my name is Harrison Coker and I'm a PhD student in the Soil Chemistry and Fertility program at Texas A&M University. And as part of my research, I am very interested in how plants will take up these metals because as we've seen, that's definitely an important aspect of growing plants in lunar simulant soils and what we expect to find once we get to the moon as well. So as we can see, the lunar surface materials have a lot of toxic metals that could be present and bioavailable to an extent that is going to impede the plant's productivity and also potentially reduce its biological longevity. But the issue is that almost no nutritional data exists from lunar regolith grown plants. And so as Moon Village Association is looking forward to sort of thinking about how we're going to feed astronauts and how we're going to use soil as a substrate to grow plants, we started getting the idea that if we don't really have that knowledge available to us from lunar simulants, that we can actually build a global plant nutrition database. The crazy thing about this is that it doesn't exist either. And so what we're finding is that if we need to create a proxy for lunar surface minerals while also doing important science right now, we can start constructing and compiling plant nutrition data for the first time on a global basis across the world. And so that's exactly what we're doing. And so far, we have built a database with around 70 studies that contains over 5,500 data points on all of the macronutrients, micronutrients, rare earth elements, and toxic metals that have been found in plant tissues growing across the world. And we really want to have a focus on obtaining all of that extreme environment data that we can, because we think this might be most relatable back to how plants are going to be growing in the lunar substrate. And so it's important to remember that plants will take up all elements that are a part of the soils that they grow in. And so we can use some GIS mapping of the high resolution data that we have here on Earth, such as we see in the left figure, and sort of relate back to how the plants that we have found in our studies and in our overviews are relating to those concentrations in the soil, and then project that onto the moon and say, hey, we're interested in the South Pole, or we're interested in this specific region. We can pull up some of that elemental data that we've obtained from satellites and sort of start thinking about, well, what plant groups are going to be most productive in these soils, and which ones are going to be able to withstand having all of these metals present in the substrate. And with that information, we really hope to get an idea of not only what might be toxic in these plant materials, but also what nutrients might be deficient, as that's going to also be an important aspect of being able to have foods that are nourishing once we start going off Earth. Thank you.
The last working group aims to examine the feasibility of growing plants in lunar regolith. We are looking at if these plants are toxic and their accumulation of certain metals and minerals above daily limits that may put astronauts at risk. We want to know if this can contribute to disease, but we also know that spaceflight causes many changes in the body. So we want to use the studies previously discussed to examine the relationship to genes that interact with these chemicals. We hope that this will lead to discussion for potential countermeasures, such as plant bioengineering to limit accumulation or various regolith amendments. Through this work, we looked at previous studies to evaluate both regolith and simulant-grown plants, as well as plants grown in harsh environments all over the world. We are using this information to build a database of studies of plants grown in these conditions. We want to know how these chemicals accumulate in toxic amounts in various plant species and in these various environments. And lastly, use the astronaut data we have to understand how spaceflight may require further considerations for toxicity and disease.
So thanks everyone for attending today's webinar regarding Lunar Agricultural Sustainability Group. We have to thank many different people that are weekly working on this project. If you are interested, you should definitely join the MBA. Here's the link. And if you have any further questions, feel free to send me an email to myself. Thank you very much and hope to talk to you soon.